Alright, let's get our clone on. So most of their lives, cells hang out in this limbo period called interphase, which means they're in between episodes of mitosis, mostly growing and working and doing all the stuff that makes them useful to us. During interphase, the long strings of DNA are loosely coiled and messy, like that dust bunny of dog fur and laundry lint under your bed. That mess of DNA is called chromatin. But as the mitosis process begins to gear up, lots of things start happening in the cell to get ready for the big division. One of the more important things that happens is that this little set of protein cylinders next to the nucleus called the centrosome duplicates itself. We're going to have to move a lot of stuff around in the nucleus, and that's going to be regulated by these centrosomes. The other thing that happens is that all of the DNA begins to replicate itself too, giving the cell 2 copies of every strand of DNA. To brush up on how DNA replicates itself like this, check out this episode and then come on back. Now the cell enters the first phase, or the prophase, when that mess of chromatin condenses and coils up on itself to produce thick strands of DNA wrapped around proteins. Those, my friends, are your chromosomes. Instead of dust bunnies, the DNA is starting to look a little bit more like dreadlocks. And the duplicates that have been made don't just float around freely, they stay attached to the original, and together they look like little X's. These are called the chromatids, and one copy is the left leg and arm of the X, and the other copy is the right leg and arm. Where they meet in the middle is called the centromere. Just so you know, these X's are also called chromosomes, sometimes double chromosomes or double-stranded chromosomes, and when the chromatids separate, they're considered individual chromosomes too. Now, while the chromosomes are forming, the nuclear envelope gets out of the way by, completely disintegrating. And the centrosomes then peel away from the nucleus, start heading to the opposite ends of the cell. As they go, they leave behind a wide trail of protein ropes called microtubules running from one centrosome to the other. You might recall from our anatomy of the animal cell that microtubules help provide a kind of structure to the cell. And this